Hi, hello, good morning everyone. I'm Dr. Harika Kishore. On behalf of Vista's Learning, I'm here as your biology educator. V-Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the live classes, pre-recorded sessions, study materials, concept videos, animated videos, assessments, and you're also going to get this lecture notes in the form of PDFs. Get your acquaintance with all the channels made available for you and subscribe to the channel Vista's Learning Class 11 and 12. If you find this session useful and interesting, make sure to click on the like as well as share this session with your friends. And also click on the bell icon so that you are going to get all the upcoming video notifications. Saying that, let's start today's session. So, we have been discussing about the chapter and that is called as molecular basis of inheritance, which comes under the unit genetics and evolution. Let's proceed into a very important process and that is called as translation. So up to now, we have been discussing about the molecular biology of the central dogma where replication followed by transcription and followed by the translation. We are very well known that the genes will express in terms of a protein which are further going to show its biochemical response. That means function. So as we already know that all the genetic information will be stored in the DNA, which makes its own copy, replica, by replication. Now, that information, that is genetic information, which is stored in the DNA will be copied into mRNA and this mRNA initially it will be present in the form of hnRNA, heterogeneous nuclear RNA, where it will undergo a series of the processings where splicing, capping, tailing takes place finally to produce a processed mRNA or mature mRNA which will cross through the nuclear pore and it will reaches to the cytoplasm from nucleus. So once we got this mRNA, will get protein. So this process is called as transcription and this is translation. So now we are here where we got mRNA, all the information which is necessary to produce protein, and we well know that proteins are made up of amino acids. Amino acids are the building blocks of protein. So what is an order or the sequence by which this amino acid has to connect by forming peptide bond, and ultimately it leads to the complete structure of the protein that is decided by the information or the codons which are present on the mRNA, right? So we are discussing about this process that is called as translation. Let's move to the translation. As we already said that translation refers to a process of polymerization, that is synthesis. Synthesis of what? Synthesis of protein of amino acids to ultimately form polypeptide. Polypeptide is nothing but your protein. So it's a polymerization of the amino acids. As I already said that each and every amino acid will bind by the means of peptide bond, and which amino acid has to come in which order, that sequence of the amino acids will be decided by the codons which are present on the mRNA. Now, the order and sequence of amino acids are defined by the sequence of the bases, that is the codons which are present on the mRNA. That is why we can say that whatever the information, that is the genetic information, is stored in the DNA that is copied onto the mRNA, that means now the mRNA is representing the genetic information. Now, by using the codons which are present on the mRNA, we get the order as well as sequence of the amino acids that are going to be present in the structure of the protein. That is all about translation. Now, if you look at the each structure of the amino acid, we can see a functional group, hydrogen, amino group, and carboxyl group. And it is always between carboxyl group of first amino acid and amino group of second amino acid, the peptide bond formation takes place, where one of the hydrogen from this and OH from this will get removed in the form of water molecule and leads to the formation of peptide bond. What is that bond? CONH bond. This is called as peptide bond. So, by forming peptide bonds, the amino acids which are present in the cytoplasm are that order and sequence by which this amino acid has to come and attach. That is nothing but polymerization. It is going to synthesize. And as we already know that the proteins are made up of amino acids, and these amino acids has to be carried by one acceptor molecule, and that is called as tRNA. By using its anticodon, as we already discussed about the structure while we are studying about the structure of the RNA, tRNA particularly, transfer RNA, we mentioned about the presence of arms or hands. At one end, the tRNA contains anticodon arm, and other end it will have an acceptor arm, where this acceptor arm will bind to a particular amino acid. Which amino acid? That will be decided by the codons which are present on the mRNA. How this tRNA is going to bind to the mRNA? Because the codons and anticodons are complementary to each other. They will form a hydrogen bonds, and thereby there is a link that is going to happen between tRNA, which is carrying amino acids, to the site of the protein synthesis. Which is the site of protein synthesis? That is ribosomes. Now, let's move forward. So here, as we already said that, for the protein synthesis happen, there are some prerequisites. We require ribosomes, we require amino acids, because amino acids are the building blocks of protein. We require mRNA, which comes from transcription, and we require tRNA. And also magnesium ions, which is useful for dissociation and association of the larger subunit as well as smaller subunit of the ribosome. So if you look into the ribosome, these are the site at which the photosynthesis, at which the protein synthesis happens. So here the ribosomes, as we already know, it is of two types. One is 70S ribosomes and other one is 80S ribosome. So we have larger subunits as well as smaller subunits. The larger subunit in the 70S is 50S and smaller subunit is 30S. Similarly, the larger subunit in the 80S ribosome is 60S and smaller subunit is 40S. So this smaller subunit will get bind to its larger subunit, and that binding, that is association and dissociation, is under the control of the concentration of magnesium, which is very, very important. And that association can be seen during the protein synthesis, and they will get dissociates when the protein synthesis is finished, that means completed. Now, so we have ribosomes, we have amino acids, we have tRNA as well as mRNA. Now, let's proceed towards the process of protein synthesis. Before the process of protein synthesis actually begins, there is a need for the activation of amino acids. Why do we activate this amino acids? Because the amino acids which are present in the cytoplasm, they will be present in an inactivated form. First, they has to be charged. We can say activation of the amino acids. Then we already know that tRNA based upon the anticodon. So it can code, it can read actually the codons which are present on the mRNA, and to that particularly it will bind to specific amino acid. That means the charged activated amino acids will come associate with a corresponding tRNA, and this is all will happen before actual the process of protein synthesis. This we can say that these are all prerequisites. Now, so the amino acids are joined by a bond. Just now we said that, we mentioned each and every amino acid will combined together, will join together, will link by the formation of the bond, and that bond is called as peptide bond. The enzyme presence is also needed to accelerate the process of formation of peptide bond, that is peptide bond formation, and the enzyme which is needed for the peptide bond formation is peptidyl transferase enzyme. What is it? Peptidyl transferase. Now, amino acids are joined by a bond, and that bond is called as peptide bond. That is CONH, which is formed by the carboxyl group of the first amino acid with the amino group of the second amino acid. Now, which is known as peptide bond. As I already mentioned, peptide bond is CONH bond. Formation of the peptide bond requires energy. It requires energy, and therefore, in the first phase itself, that is, we can consider it is a prerequisite. Before actually moving into the protein synthesis, first the amino acid has to be activated, and for that activation it requires energy, and that energy comes from ATP. Itself, amino acids are activated. Activation of the amino acids takes place. Charging of the amino acids takes place. So, amino acids are activated in the presence of ATP and linked to the correspondent cognate tRNA. And this process, what is the process? Activation of amino acids by using ATP, and that will associate with its tRNA, specific tRNA. All this process, we call it as a charging of tRNA, or specifically to be mentioned, it is amino acylation of tRNA. So, amino acylation of tRNA happens in the first phase itself before it is going to proceed towards a protein synthesis, to be more specific. So, this process, to be more specific, it is an amino acylation of tRNA. Let's take a look at this amino acylation of tRNA, how it is going to be takes place. So, as we already said that the protein synthesis is happening in the cytoplasm of the cell, and in the cytoplasm of the cell we have a pool of amino acids, and this amino acids will be present in an inactivated form. First it has to be activated, and for that it requires energy. That energy comes from ATP. So here, amino acid activation, right? For that, amino acid combines with ATP. For this, it requires enzyme, and that enzyme is amino acyl synthetase. Now, which leads to the formation of a complex plus pyrophosphate. What is this complex? This complex, we call it as an amino acyl adenylate complex. See, this is all about the activation of amino acid in the presence of ATP as an energy, in the presence of an enzyme, and that enzyme is amino acyl synthetase. Amino acyl synthetase, it will form a complex, and that complex is called as amino acyl adenylate complex. right? Now it has to associate. See, once the activation is done, we are at here. The first phase itself, amino acids are activated in the presence of ATP, activation of amino acids in the presence of ATP and linked to their respective cognate tRNA. Now, this complex plus tRNA, because it has to associate with a specific respective corresponding tRNA. So here it will lead to the formation of amino acyl tRNA complex. Amino acyl tRNA complex plus AMP plus enzyme, and this enzyme will be used again and again. So here ultimately it leads to the formation of amino acyl tRNA complex. So that is what called as a charging of tRNA or amino acylation of tRNA. This amino acylation of tRNA is nothing but the formation of the complex, and that complex is amino acyl tRNA complex. Now, moving ahead. So here, what tRNA is doing? tRNA has an anticodon by which it can read the code which is present on the mRNA, and based upon the code which is present on the mRNA, it will identify activated amino acid, and that activated amino acid will come and attach to the acceptor arm. Let's look at the structure as we already discussed. I'll draw here. Amino acid. So that is what this arm is considered as an acceptor arm. And as here we can find anticodon presence, this arm is called as an anticodon arm. So this anticodon arm can complementary made a base pairs with the codons which are present on the mRNA. So here, if two such charged tRNA, what do you mean by charged tRNA? Nothing but amino acyl tRNA complex. Two amino acyl tRNA complex will are brought close enough, see, are brought close enough on the mRNA, the formation of peptide bond between them means amino acids will happen, and it will carry out energetically by using ATP. Now, the presence of the catalyst would enhance the rate of the peptide bond formation, and that catalyst or the enzyme which is useful for the formation of peptide bond formation is peptidyl transferase. Peptidyl transferase. Now, the cellular factory responsible for synthesizing proteins is a ribosome. Ribosomes are the site for protein synthesis to take place. And they are of two types. Just now we said that they are 70S ribosomes, where we find it in prokaryotic cell as well as in the eukaryotic cell. 70S type of ribosomes are found in eukaryotic cell. Where do we find? We find it in the mitochondria as well as in the chloroplast. Whereas 80S type of ribosomes are exclusively present in the eukaryotic cell. So here we can say that the 70S type of ribosomes can be seen in both prokaryotic cells and eukaryotic cells, whereas the 80S type of ribosomes are exclusively present in the eukaryotic types of cells. So eukaryotic organisms have this 80S type of ribosomes, where it will show larger subunit as well as smaller subunit. Larger subunit is 60 and smaller subunit is 40S. Now, moving forward. So, as we just now said that the ribosomes consisting of a structural RNA as well as so many different types of proteins are also present. Around 80 different proteins can be seen in the ribosomes, and it is an inactivated state. It exists as before the photosynthesis takes place. Before the protein synthesis takes place, this ribosomes exist in an inactivated form. We have larger subunit, we have smaller subunit. We have smaller subunit. And in its inactivated form, it exists as two subunits, that is larger subunit and smaller subunit, that is a larger subunit and a smaller subunit, right? When the smaller subunit encounter an mRNA, see, consider this is an mRNA which runs from 5' end to 3' end. When the smaller subunit encounter the mRNA, the process of translation of the mRNA to the proteins begins. Then, first always the smaller subunit will attach to the mRNA, then the larger subunit will come and bind. And this association is very important. The larger subunit as well as the smaller subunit of the ribosomes has to be associated while the protein synthesis is in progress. Now, just we said that they are two sites. See, consider this is an mRNA and this mRNA is encountered by the smaller subunit of the ribosome and the larger subunit will come and attach. Here in the larger subunit, we'll find sites. So, we will have exit site, peptidal site or polymerization site or donor site, and a acceptor site or amino acyl site. So here they are two sites. Majorly importance are given to these two sites because this site is for exist of the used up tRNA, and this tRNA will leave the ribosomes to carry an amino acid, which is a specific amino acid if it comes again by the, which is decided by the codons that are present on the mRNA. So this site is particularly for exit of the tRNA, that is the tRNA once it leaves its amino acid. Once the tRNA enters, it will get the amino acid. Once it is leaving, it leaves that amino acid and leave the site of the and will be added back to the cytoplasm of the cell. So this is the site for exit of the tRNA. Therefore, the importance has been given to the two sites, and that site or one is P site and other one is A site. So here, in the larger subunit, where do we find the presence of the sites? The sites will be present towards the larger side of the larger subunit of the ribosome, and the smaller subunit of the ribosome will first encounter to the mRNA. Then after, the larger subunit of the ribosome will come and combined with this. Right? Now, for subsequent amino acids to bind and to thus be close enough, these two sites will be close enough to each other for the formation of peptide bond, So, in this site, whatever the amino acids that has been chained up in the formation of polypeptides, that will be transferred to the A site. So, that is what this P site as well as A site will be very close to each other. Now, the ribosomes also act as a catalyst. 23S, that is a subunit or RNA in the bacteria, and that enzyme is called as ribozyme. Ribozyme is a ribosomal enzyme which is useful for the formation of peptide bonds in prokaryotic organisms, that is bacteria. Now, so here, a translation unit, see, while we are discussing about transcription, mRNA, DNA, and this is called as transcription. While we are discussing about this process, we have mentioned about the transcription unit. This is the transcription unit where we'll have promoter, right? Structural gene and terminator, right? So, yeah, terminator. So, similarly, we have transcriptional unit also. A transcription unit in mRNA is a sequence of RNA that is flagged on either side, just like the structural gene which has on either side, on one side it has promoter and the other side it has terminator. Here also the sequence of the RNA will have stop codon on one side and that means start codon on one side and stop codon on another side. So flagged by the start codon, which is represented by methionine code, that is AUG, and the stop codons which codes stop codon and code for the polypeptide. Which code for the polypeptide? RNA sequence. That is a sequence of RNA. On either side, it will have start codon and stop codon. Now, so here we have seen transcription unit. Similarly, here it has flanked with the promoter and terminator. Similarly, here it is. It always polymerization runs from 5' to 3'. So here we can see start codon and stop codon. Here is a sequence of RNA which code for polypeptide. So, a mRNA, this is a mRNA also have an additional sequences other than the start codon sequence of the RNA and stop codon. It will have an additional sequences that are not translated, which will not be translated and or refers to as it is not being translated, it is referred to as untranslated regions, UTR. And these UTRs are present on both 5' as well as on the 3'. So, we find it either on 5' that is before stop codon and on 3' end that is after. This is stop codon, no? After we find this UTR. So, the presence of these UTRs are required for efficient translation process. Now, this is all about the translational unit. For initiation, the ribosomes binds to the mRNA. As we already said, the smaller subunit of the ribosome go and bind to the mRNA at the start codon. And start codon is AUG, which code for the methionine, that we already know. And it is recognized only by tRNA, and that tRNA, as it is initiating the process of translation, we call it as an initiator tRNA, and that initiator tRNA is nothing but F met tRNA, that is formal methionine tRNA, which is acting as a initiator tRNA. And here is a point. Every amino acyl tRNA come and bind to the A site which is present on the larger subunit of the ribosome, except the first, that is F-met tRNA, which is going to fix to the P site and leaving the A site vacant. and that vacancy will be covered up with the next coming amino acid tRNA. This point is very important to be noted, and the remaining is very easy to understand. So here, the ribosome, this is how the ribosome, while the procedure, that is the elongation of the translation happens, this is how it will look like. Here we have another site that is called as exit site, as I already told that this exit site is useful for the removal of the used tRNA. See, this tRNA carried amino acid carrying tRNA. It is coming and going to fix to the A site. Then there is a shift of how many codes? Only one code at one time. So if you think that this is an mRNA, first it is fixed with the smaller subunit of the ribosome. Then larger subunit of the ribosome is coming and fixing like this. So every time the ribosome is going to move ahead by one codon. Every time it is moving ahead by one codon. So it will move by one codon. In that way, where whatever the polypeptide, that will shift from P site to A site. That happened, and the another site will come and fix to the A sites. So this shift will be happening in such a way the entire mRNA which is containing the codon has been read with an anticodon that are present on the tRNA. So in that way, every time it will take a move of each codon, each codon, each codon. So initially the mRNA is towards this side, and towards the end of the process the mRNA has been read off and the polypeptide chain has been increased. So this is how, see, during this stage, complexes composed of amino acid linked to tRNA. Amino acid linked to tRNA. Initially, this F-met tRNA will go and fix to the P site, but after coming amino acid tRNA will go to the A site. Meanwhile, the peptide bond will form in between these amino acids, and this chain will shift its position. Now leaving every time A site vacant. So this is P site and this is A site. Shift happens from P site to A site, and every time this A site will be ready for the next attachment. So this is how happened. See, now this increased is a growing polypeptide chain. So look at here, during this stage, complexes composed of amino acid linked to tRNA subsequently binds to the appropriate codon. Which is present on mRNA. As it is having a complementary base pairing with the anticodons that are present on. These are anticodons. Anticodons are present on tRNA, and these are codon which is present on mRNA, and this codon will decide which amino acid has to be bring by the specific tRNA. Now, the ribosomes moves, as I just now said that ribosomes every time move one codon ahead. The ribosomes move from one codon to codon. It moves from codon to codon along the length of the mRNA. Amino acids are added. See, amino acids has been added and added one by one, translated into polypeptides. This is your polypeptide chain. This is your polypeptide chain, still growing by shifting from one codon to the another codon. And this is directed, and this is dictated by DNA, because DNA is a site where the genetic material is going to store, and it is represented, just represented by the mRNA as it get translated. So first DNA, then mRNA, that translate into protein. So the formation of this protein, the information is given by the DNA, therefore it dictates, and it is copied on to the mRNA, therefore it represents. So dictated by DNA and represented by mRNA. At the end, a releasing factors binds to the stop codon. Once this ribosome move ahead by one codon which are present on the mRNA, once it reaches to the stop codons, either of the 3 stop codons, there by using the releasing factor binds to the stop codon, terminating. It comes to the end of the translation process and it will releasing the complete polypeptide from the ribosomes. So once the protein synthesis is done, all this will get dissociate. Subunits of the ribosomes will get dissociate, and even these ribosomes will get dissociate from the mRNA, leaving the growing, that is a fully formed polypeptide chain free. Now this polypeptide will show some of the again processing where it will fold itself to form a complete structure of the protein according to its particular function to be performed by that particular protein. Maybe this protein is going to act as an hormone, maybe this protein is going to act as an cofactor or enzyme, anything based upon its need to be performed, based upon its function. The structural changes will happen to this polypeptide chain, right? Yeah, that is it. So today's session will discuss all about the translation, how the protein synthesis is going to happen by using tRNA, mRNA and larger as well as smaller subunit of the ribosome. We have seen about the detailed structure of tRNA as well as how the activation of the amino acids takes place and how it is going to bind with the tRNA and finally leads to the formation of amino acid tRNA complex, right? So let's meet up in the next class where we are going to discuss about the regulation of genes. Until then, keep learning with We Learning. Thank you.